We all know that one light switch can control many light bulbs. A simple process, right? But how could this relate to operons? Jacob, Sánchez and Monod, three key scientists who discovered the LacZ operon in 1960, put it simply. Operons are a cluster of genes under the control of a single promoter, like light bulbs under the control of one switch. Operons are a well-known feature of bacteria and archaea genomes, enabling the efficient and coordinated expression of 2 to over 10 usually functionally related genes. In 1993, operons were discovered in the eukaryotes C. elegans, followed by flatworms, and more recently in Drosophila. Genes within operons are transcribed as a single polycistronic mRNA from one upstream promoter. In some eukaryotes, such as C. elegans, the polycistronic mRNA is transpliced into monocistronic mRNA and individually translated by ribosomes which scan for the first start codon. However, prokaryote mRNAs remain polycistronic, and so ribosomes must select for the correct start codon. Shine-Delgado motifs help ensure the efficiency of this process. These motifs are 5' to the initiated codon on mRNA and can base pair with the anti-Shine-Delgado sequence of the 16 subunit ribosomal RNA to help assemble the translation initiation complex. The high Shine-Delgado presence within operons demonstrates its importance for their translation. Let's consider the very first operon discovered, the LacZ operon in E. coli for lactose metabolism. The LacZ operon is composed of LacZ encoding beta-galactosidase for lactose breakdown, and a mutated version of this allele, Z- produces the enzymatically inactive protein CZ. LacY encodes galactoside permease for lactose transport, and LacA encodes transacetylase. Jumping back to 1942, Monod had discovered bacteria switched to metabolize lactose for energy when glucose was depleted, termed dioxic growth. He also found allolactose is synthesized in the presence of lactose and was like an inducer for beta-galactosidase. Following this, the LacZ structural genes were identified to be inhibited by a repressor synthesized from a regulatory gene. It was then hypothesized that an operator was sensitive to this repressor and repression could be relieved by an inducer. This brings us to 1960, where the scientists sought to study how operators intervened with gene expression by studying operon mutations. The scientists obtained diploid E. coli for the genes I, O, Z, and Y through conjugation, whereby a sex factor with a small fragment of bacterial chromosome was transmitted to the recipient bacterium. With different recombinations, they studied how different mutations impacted proteins produced in induced bacteria who had inactivated repressors due to the presence of lactose, or uninduced bacteria where repressors are active due to the absence of lactose. They measured beta-galactosidase, protein CZ, and the permease as a percentage of what was obtained for the allele of induced wild-type bacteria. Sometimes an excess of galactosidase was recorded simply because of the presence of several FLAC factors per chromosome. Now, let's look at the scientist's constitutive operator mutant. Here, the operator had lost sensitivity for the repressor, resulting in the constitutive expression of LacZ and Y. Let's look at the data that they obtained for a diploid bacterium of this genotype. Notice that the uninduced bacteria had 110% synthesis of galactosidase, no protein CZ, and 50% permease. However, induced bacteria synthesized all three proteins. So, for heterozygotes, the constitutive operator mutation is dominant only for genes in the cis position, that is, the same side of the mutated operator. For instance, the mutated Z allele is in position trans relative to OC and remains only inducible. They also looked at a second recessive mutant strain, which inactivated the operon. They found that this mutation was also between loci Z and I. The scientists concluded that operons are composed of an operator and the structural genes it coordinates and is sensitive to a repressor. Repression may be at the level of genetic material or at a cytoplasmic version of the operon. However, what did the researchers not account for? They did not know if the repressor was protein or RNA. However, in 1966, the LacZ repressor was identified to be a protein which binds to the operator. Following this, allolactose was found to bind to the repressor, decreasing its affinity for the operator, enabling transcription of the LacZ operon. The researchers also did not account for activators of operons. Considering the LacZ operon in glucose starvation, cyclic AMP was synthesized and binds to a protein which binds to DNA, enhancing RNA polymerase's ability to transcribe genes. Jacob and Monod won the Nobel Prize in 1965. But looking at the bigger picture, the regulatory mechanisms of the LacZ operon does not just apply to E. coli. In a mouse, the LacZ operator repressor system was successfully transferred to control tyrosinase gene transcription. Like the light switch, this bacterial switch may even eventually be used to switch on and off reversible models of human disease.